Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between Dr. Paloma Giangrande, Editor-in-Chief of Molecular Therapy, Nucleic Acids, and Chief Technology Officer at 11 Therapeutics, and Dr. Marcin Kortylewski, Professor, Department of Immuno-Oncology, City of Hope. They will discuss a recent article published in Molecular Therapy, Nucleic Acids by Dr. Kortylewski and colleagues titled Oligo-PROTAC Strategy for Cell-Selective and Targeted Degradation of Activated STAT3. If you enjoy today's conversation, you'll also enjoy the upcoming ASGCT Policy Summit in Washington, D.C. on September 23rd and 24th. This can't-miss event brings together policymakers and gene and cell therapy experts, including FDA leaders Julie Tierney and Dr. Nicole Verdun, to discuss the latest policies impacting this rapidly evolving field. Register now at asgct.org/policy summit for invaluable insights on navigating the regulatory landscape. That's the ASGCT Policy Summit, September 23rd through 24th in Washington, D.C. Virtual attendance options are also available. And now, Drs. Paloma Giangrande and Marcin Kortylewski.
Hello, everyone. I'm very excited to be here today with Dr. Marcin Kortylewski, professor at the City of Hope. And as you know, we're here today to discuss Dr. Kortylewski's new paper that was recently published as of March 2024 in Molecular Therapy Nucleic Acids. And so with that, welcome, Marcin Kortylewski. Dr. Kortylewski, it's a pleasure to have you here. Before we start, I want to say that I have known Marcin for the longest time. We first met back in 2007 when he was a postdoc at the City of Hope and was already working on the research that he's published in molecular therapy recently, having an interest in STAT as a target, STAT3 in particular as a target, for cancer immunotherapy as well as immunotherapy applications. He began his work really early on, stayed focused on this particular target, and has come up with various approaches over the years to go after this target for these particular indications. So again, Marcin, welcome. And why don't you take the first couple of minutes to introduce yourself to our audience?
Sounds good. Thank you so much, Paloma. Thank you for the invitation. It's a pleasure to be here. And let me just say a few words about myself. Thank you for a beautiful introduction. I was trained in molecular biology originally, then jumped the ship and focused on tumor immunology, which turned out to was an opportunity to use a lot of skills in science signaling game. And then I think I got relatively early associated with certain type of signaling, which was JAK-STAT signaling, and that became kind of an obsession over the last 20 and more years, where we really focused our research on first understanding role of STAT3 in immune cells, and specifically in cells that present antigens, and then started searching for ways to target these specific molecules. And it brought me somehow to the field of oligonucleotide therapeutics, and that's how we met. And I continue to be really interested in employing oligonucleotides, DNA, RNA molecules, for the purpose of cancer immunotherapy, where I think they have still a huge area of opportunity that have not been fully explored yet.
And so you've already mentioned your interest in STAT3. Can you tell us a little bit more of why STAT3 in particular is such an interesting target for cancer immunotherapy and cancer therapy in general?
Yeah, so when I was entering the field, I mean, my interest in STAT3 signaling was also driven by the fact that it seemed very simple or deceptively simple in contrast to other signaling pathways that require a lot of multiple molecules and scaffolds to really transmit the signal from the membrane to the nucleus. In case of JAK-STAT, it seemed to be a combination of a receptor with a kinase and this multitasking STAT protein that could get the signal at the membrane level, then travel through the whole cytosol, get to the nucleus and regulate gene expression. So it looked really as a simple pathway to understand where one protein plays most of the functions. It turned out to be more complicated than that. There's a lot of regulation that needs to happen to make this pathway work. I think that STAT3 became really attractive and searched protein around the end of 90s when its potentially oncogenic functions have been described in cancerous cells on transformed cells. So that kind of skewed a lot of attention, and it continues until now on STAT3 role in cancer cells. And it's justifiably so. It plays a very important role in hematologic malignancies. And many of these malignancies really depend on STAT3 for survival, and that can be a major function that can be exploited therapeutically. It's more complicated in solid tumors. So they very often use STAT3 as one of many alternative pathways to promote their survival. So it's more difficult to target STAT3 and see long-term therapeutic or curative effects in context of solid tumors. So STAT3 as an oncogene in cancer cells is not always the most reliable target. So for us, and I think for STAT3 targeting field, I think it's really important to look at this molecule and its role in immune cells, and specifically in cells that become associated with tumors when they grow. And because in immune cells, I mean, basically the playbook remains solid and remains unchanged during the evolution of a tumor. The signaling pathways that operate in immune cells and genetically stable normal cells remain unchanged. So STAT3 performs very important functions in myeloid cells associated with tumors, as well as in T cells. But its role in myeloid cells is probably the most attractive to explore for therapies, for cancer therapies, since it seems to be a major brake on antigen presentation, on secretion of pro-inflammatory immunostimulatory cytokines, interferons as well as IL-12. It basically dampens the ability of cells to present antigens, especially on MHC class II complexes, and triggering Th1 helper cell activity becomes really difficult, which is typical for immune suppressive environment in the tumor. So STAT3 has this clear role of a brake in antigen presenting cells that can be dendritic cells, but also macrophages and microglia in context of brain tumors.
Oh, sorry, go ahead.
Go ahead.
Sorry. No, it seems like you're touching on these cell-specific roles of STAT3. And oftentimes when most people think of cancer therapies or cancer immunotherapies, they associate them with chemotherapy, for example, and other types of therapies that are very non-specific. Now, are there any safety issues of targeting STAT3? Can you target STAT3 globally? Do you need to go after a tissue and cell-specific approach to STAT3 inhibition for cancer immunotherapy? And if so, how would you go about doing it, I guess?
Right. So that's a very, very important issue. And so it's easy. And I mean, it happened many times that STAT3 has been associated with this oncogenic role, has been seen as a negative player in context of cancer immunotherapy as a potential target. But it's more complicated than that. I mean, it's the role of STAT3 in T cells is very often positive, and it's necessary for their expansion in the context of immune responses. CAR T cells benefit from STAT3 activity, so there are approaches which actually bring receptors that can activate STAT3, like IL-23 receptors, into CAR T cells, basically to make them more effective and to let them expand better. So globally targeting STAT3 in context of cancer immunotherapy can be quite counterproductive, or at least can balance out the positive and negative effects. On the other hand, STAT3 targeting and STAT3 inhibition, initially we were quite worried what could happen with STAT3 blocking, because the original studies eliminating STAT3 gene in mice were showing that it can be embryonically lethal. And then it took about 15 years to see first studies coming from NIH, Mulholland's group, showing that first deletion of or functional deletion of STAT3 can lead to hyper IgE syndrome or JOPS syndrome, which was related with defective Th17 T cell biology and defective clearance of microbial and fungal infections from the skin, which can be over a long time. It could become lethal, but this is not early lethality. And then we also learned about gain of function mutations within STAT3, that it's rarely related with generation of cancers. So some lymphomas, NK lymphomas, can be generated in this way, but there's a lot of disruption in context of immune system, a lot of autoinflammatory diseases that can arise as a result of gain of function mutations in STAT3. So blocking seems to be a little bit less problematic than constitutive activity of STAT3, which in context of human immune system biology.
Yeah. And so you have gone, since the very beginning, you have gone after some very interesting approaches to enable cell-specific or cell-selective targeting of STAT3. Do you mind describing some of those approaches with, that you've undertaken with oligonucleotides?
Sure, right. So we have tested initially small molecules as well, and then that was an uphill battle. So we decided that for us it's critical to deliver the STAT3 inhibitor specific to antigen-presenting cells, and these happen to be cells that are well trained in picking up debris from the microenvironment. They basically are trained to internalize extracellular DNA. Usually that would come from either microbes or mitochondrial DNA. So dendritic cells, macrophages, can really do it very effectively. And even the immature myeloid cells do it quite well. So we decided that would give us a kind of entry gate into the immune system and into the very important part of the immune system, because these are antigen-presenting cells are basically this brain of the immune system, which processes the information before initiating immune responses. So we decided to use a synthetic piece of DNA, which have been widely studied. That was beginning of 2000s. There was a lot of trials using unmethylated CpG motifs in DNA, synthetic DNAs, pioneered by Art Krieg, who found these motifs in antisense molecules. And that has been seen first as an obstacle for antisense molecule development, then was separated into immunotherapeutic approach that could be used in context of oncology. So we have used basically such short stretch of DNA, attached that to a STAT3 siRNA, and then we were really surprised to see that these much longer molecules with triple the size of the original DNA were able to be internalized by target cells quite effectively within less than an hour. And that could be also done in vivo in mice, allowing us to enter those antigen-presenting cells. We could inject these molecules into the tumor, label them fluorescently, and follow them when migrating with those antigen-presenting cells, with dendritic cells and macrophages, into the tumor-draining lymph node, seeing basically what happens, and confirming that these activated antigen-presenting cells can also interact with T cells and trigger anti-tumor immune responses. So that was our proof of concept study.
Yeah, no, that's great. And do you understand the specific molecular mechanism of CpG uptake in these cells? Is it via a receptor that plays a key role? Yeah. If you can elaborate on that.
Yeah, we spent some time trying to figure out the mechanism of uptake and also the mechanism of processing of this molecule later. The mechanism of uptake turned out to depend not on the receptor that we originally targeted, because the toll-like receptor 9 that recognizes those CPG, unmethylated CPG motifs, is in an active form, is present in the endosome. It rarely pops up on the membrane, and if it does, it's not properly processed and it's not active. So the initial interaction between this chemically modified DNA, phosphorylated DNA, occurs between this molecule and scavenger receptors on the surface of target cells. So that's a broad family of receptors that is originally described to bind and deliver oxidized lipids. So it's a part of lipid metabolism that is also an important part of how these myeloid cells survive in a context of tumor microenvironment. There's plenty of these receptors, so it is a very effective system of uptake that we have not managed to saturate or downregulate as a result of repeated treatments, which allows for an uptake of a large amount of the oligo. But importantly, when it gets in the endosome and it triggers toll-like receptor 9 activity, it turned out, to our surprise, that has an effect on accelerated release of the content of the endosome into cytosol. So that was a big kind of a boost for our initial proof of concept strategy, because the stability of this proof of concept molecule was relatively low. It had to be processed, delivered quickly. So this quick release from the endosome within the first 2 hours after uptake was a great way to enhance and improve the concentration of the siRNA in the cytosol.
That's great. And we already alluded to this a little bit at the very beginning. You've gone after STAT3 inhibition with various different modalities, or taking various different approaches. Can you describe a little bit these different ways that you've gone about it with the different modalities like RNAi, antisense oligos, decoys, given that the subject of the paper that we're discussing today is a decoy strategy? If you can comment on these different strategies, and why did you land on the decoy strategy? So a little bit of your thoughts on these different ways for inhibiting STAT3.
Sure. So we started with siRNA, but the original design that we used relied on a cleavable molecule, and which this design worked really well in local setting, but we had issue with a molecule being cleaved too early when injected intravenously, which definitely happened even more. It was even more likely to happen in the context of human serum. So we started to explore alternative strategies that we wanted to go away from RNA for the time, moving towards antisense strategy, and then in parallel also decoy DNA. So these are two, as antisense strategy, similar like siRNA, will downregulate STAT3 at mRNA level, so it takes a while, but it can be very effective and even longer term effect. Unlike siRNA, this is not an enzymatic process, so it might require a little bit higher concentrations.
Yeah.
The target, but stability of CPG STAT3 antisense molecules was great. So we could definitely see long-term knockdowns. And we also found that these molecules don't disappear intracellularly. They can also end up in exosomes. They can be actually shedded by the target cell in encapsulated exosome encapsulated form that we can collect and reuse if we want to, which was unexpected. But uncleavable molecules basically can have a second life after this initial uptake as a taken molecule, which could kind of broaden their spectrum of targets or half-life in vivo. So that was an interesting observation. And decoy, that is the subject of this paper, or this iteration of the strategy came up in this paper, is a completely different strategy, very old one, which have been generated basically out of the need to detect transcription factors. So decoy molecules are basically double-stranded DNA molecules, which span over consensus sequence for DNA binding for different transcription factors. Yeah. It's, of course, very well-defined consensus sequence for STAT3, which binds also STAT1. So there's additional activity here. And decoy molecules, I mean, of course, as DNA have been stable, and there have been an early work done by Jennifer Grandes, which has used the double-stranded DNA decoy for STAT3 in phase 0 study, showing that it can be relatively safe and confirming also the effect on the target. So that kind of brought our attention to the molecule. The missing part was how to deliver this double-stranded decoy. So we decided to use our CpG scavenger receptor TL9 dependent uptake for that, and it turned out to work really well. We created a hairpin molecule where basically we would synthesize a single DNA molecule, creating a hairpin around the STAT3 DNA binding domain. So the interesting feature of a decoy is that it can very rapidly, after entering the cell, inhibit STAT3 by interacting with STAT3 dimers. So it doesn't need to wait until protein expression of STAT3 goes down. It can start sequestering STAT3 very rapidly. So that what attracted us in terms of potential for immunomodulation, because you would not need to wait for transcription factor expression to go down. You could see probably the effects far more rapidly.
Immediately, yeah, because you're acting right at the transcription factor level, at the protein level. So based on this, I know that you've sort of launched or started a company from the work in your lab. Is the decoy approach the one that you think it's more suited for clinical translation, or some of the other approaches, or are you combining all of these approaches for clinical applications?
I think they might find different applications in different diseases. Right now, I think the company that I started, eventually changed name to Duet Biotherapeutics, is focusing on development of antisense molecules, so STAT3 antisense, because of its high stability, its efficacy in solid tumors, which required extensive chemical modifications to make a drug solid. But I think with this new iteration of decoy molecule with PROTAC function, that would allow us to bring down protein levels of STAT3 even more, and also convert. So one of the limitations of a decoy strategy was that it really depends on interaction with STAT3 as a protein, which is driven by concentration, intracellular concentration of the decoy. Time, of course, it will be diminished. STAT3 might become released and is entering the game again. So with PROTAC function added to the decoy, we could finally have a molecule that acts early on, relatively rapidly kicks in, but it also can hold the STAT3 levels low over extended period of time.
And so before we go into the PROTAC, because I want you to define this for our audience in more detail, the decoy concentration in cells that you highlighted as a potential issue or hurdle to using a decoy as a potential clinical strategy, is that, in your opinion, the reason why to date, unless I'm wrong, but they're compared to other oligonucleotide therapeutics, we hear about RNAi, we hear about antisense, from Alnylam and Ionis. We don't hear as much about decoy drugs in the clinic. Is that one of the main reasons why we have fewer decoy drugs?
Yeah, I think that probably major kind of a cooling effect, that the fact that the decoy is a reversible inhibitor of a downstream target definitely cools people interest in decoy technology when it comes to targeting cancer cells. And most of the application would go probably after oncogenic transcription factor in cancer cells. It's a little bit different, and this is why we were focused on using decoy in context of hematologic malignancies, where we have seen that blocking STAT3 even temporarily is sufficient to induce differentiation of these cells, and that was a one-way street. So after blocking STAT3, it was not required basically to block STAT3 permanently to see the effect of initiating cell differentiation. So we studied in the context of leukemia. We have looked also in some types of lymphoma, where you can, by the combination of STAT3 blocking and triggering immunostimulation, TLR9-driven myeloid differentiation, you can see cells differentiating into antigen-presenting cells, and this is biologically a one-way street, which does not require that we continue to suppress STAT3. So in this context, decoy had a huge benefit because it could act rapidly on and trigger this transition stage. But it won't do it in the context of solid tumors, or it's more limited in terms of eliminating survival benefit in case of oncogenic targets.
And so can you go a little bit more into what is a PROTAC and why is your approach of using these Oligo-PROTAC combinations better potentially than some of the standard ways that people have or thinking about PROTACs, which is more based on small molecules and PROTAC combinations?
Sure. Yeah, I wouldn't say that we try to create something better than small molecule PROTACs, but we definitely try to develop this technology in a slightly different area, where I think small molecules with their lack of selectivity, like lack of cell selectivity, cannot perform as well. And I'm talking about basically targeting immune cells and trying to manipulate and reactivate immune system activity in context of tumors. So here you really need to go after kind of nodal points of the immune network. You can't blast signaling pathway across the board in all the different subsets of immune cells. So I think here we have an advantage. And so the PROTACs in general, this is an idea that has over 20 years. I think started by Craig Cruz lab that basically found a way to observe basically that it would be possible to use a small molecule as a bait for ubiquitination machinery for certain proteins like VHL or cereblon proteins, acting as a receptor, allowing us to bring in the whole ubiquitination machinery to protein targets. So that will basically requires two small molecules, one that acts as a bait for cereblon, and the other one that is acting as a bait for our protein of interest.
So essentially, with your approach, you're taking the intracellular protein degradation machinery and you're targeting it inside the cell to a target of interest that you want to inhibit or degrade.
That's exactly what won't happen. So it's an elegant mechanism. It's a great strategy to go after proteins where we don't have a high affinity binding site, which normally is required for development of good small molecule inhibitors that should enter some kind of catalytic site and sit there tightly, which would require a low concentration of this molecule. So here you don't need to target a catalytic site of the protein. It can be some kind of a site, away from this major catalytic site. The affinity can be lower, but the ability to bring in and ubiquitinate the protein really causes potent downregulation of the target protein. So that technology, I mean, it's made huge progress and it's in clinical trials. I'm following on trials related to immunomodulation or targeting hematologic malignancies. There are some really interesting BTK inhibitors by Nurix. There is, of course, Chimera with some STAT3 targeting PROTAC, small molecule PROTAC, with, I think, recently reported at the beginning of June in one of the congresses in Europe. I think they were reporting that it's safe and it seems to have effect on certain types of one patient lymphoma and maybe a couple of patients with solid tumors showing stable disease. So this is really very attractive. At the same time, there are some challenges for small molecule products because two small molecules bound together have relatively large size. The pharmacokinetic properties are not ideal, and the design of these products is still quite empirical. So it's difficult to predict whether product mechanism will be effective, whether it will kick in. Also difficult to predict which receptor for E3 ubiquitination complex to pick, and there are many choices. They just can have different activity in the cytosol nucleus, so that complicates the design.
And earlier, you already touched on a little bit on DNA versus RNA and the advantages of using DNA for some applications like antisense as well as decoys. There are also, so decoys in general come in two flavors, as you're very well aware. One is the DNA flavor, the other one is the RNA flavor. In your opinion, what is the advantage of DNA versus RNA? And do you see this approach being broadly applicable to other targets in addition to STAT3?
Yeah, so we have focus on DNA decoys since there's a huge collection of the potential consensus sequences that can be utilized for targeting transcription factors and also in immune cells. So it's we can go off STAT3, but we also focus on other transcription factors, for example, NF-kappa B. But I mean, DNA compared to RNA has a benefit of, on one hand, we already know the sequences. It's more stable and does not create secondary structures that would be necessary for its function. However, I would not dismiss the potential of RNA selection and generating aptamers to basically.
So if you have like a chemically stabilized RNA, like
exactly.
a 2' fluoro modified aptamer, 2' methyl modified, you think that could also have an application?
Absolutely, because that could open up a large number of additional intracellular targets which might not have DNA binding domain, but could be targeted with RNA aptamers, stable chemically modified RNA aptamers that also can be easily equipped with the PROTAC function.
That's great. That's great. And so because we're coming to the very end here, any other thoughts about your paper that you would like to highlight that we haven't touched on today that you would like our audience to be aware of?
So, yeah, I mean, I wanted basically to highlight that the potential for generating a similar oligo-PROTAC approaches for other transcription factors. And the fact that in this case, we presented that this strategy can be delivered cell selectively into antigen-presenting cells. STAT3 is a very important and excellent target in context of tolerogenic tumor microenvironment, highly immune suppressive environment. But we also work on targeting NF-kappa B, and I think this is a great potential target with where we benefit from utilizing a single consensus sequence, single decoy, and that allows us to target multiple complexes of NF-kappa B, which is far more heterogeneous than STAT3. STAT3 creates a heterodimer with STAT1, but NF-kappa B has more than 10 different complexes that could be created, which can bind to relatively similar consensus sequence. So in context of NF-kappa B promoting survival of cancer cells, and we focus specifically on B-cell lymphoma, that can be a huge advantage over, let's say, siRNA or antisense technologies that will go after just a single, very well-defined, but single target, whether it's NF-kappa P65 or P50 subunit. But it does not allow you to easily eliminate whole set of different subunits, making basically the strategy very effective.
Yeah, thank you very much, Marcin. This was a great discussion. Looking forward to reading more papers and more work that comes out of your lab. So thank you for being here. It's been a pleasure.
Thank you. It was a pleasure for me. Thank you for great questions. And yeah, looking forward to more interactions. Thank you.